It's a real honor for me to introduce Dr. Ken Anderson that you've heard so much about. He's definitely the leader in the field of multiple myeloma, and he really is so generous with his time to come here and talk to us about immunotherapy. Ken.
Thank you very much, Leif. It's really my honor to be here. I want to thank Barbara and Jack, and for that matter, all of you who are patients, because as you've heard from all of the presenters, you are our heroes and honestly the inspiration for everything we do. And all of the progress that we can celebrate here together, and hopefully some of the new lessons you take from here, remember that you're right in the middle of all of it, and without your generosity and willing to go on to trials and try new things, none of the progress would have been possible. So not only have things gotten better, hopefully for many of you in this room, all of you in this room, but you're setting the stage for generations to come. And as I like to say, and I was going to say this at the end, but I think in our world today, I don't know what you think, but I think sometimes watching the news and otherwise, I think we've forgotten to take care of each other. This is my summary. And I would say that this meeting with Barbara and Jack and the MMRF, honestly, this is, for me, really inspirational and an example of how we're all in it together, and by everybody pulling together and working for a common cause and helping each other, everybody wins. And so I want to thank you from my heartfelt thanks, but also from all the caregivers and researchers here, because it's been a team sport. And I'm not going to talk about any Boston teams because I don't want to spoil my welcome. Okay, enough of that. So I've been asked to talk about immune therapies, and you're going to hear wonderful talk about genomics from Jonathan Keats in a little bit. That's arguably one of the hottest areas in medicine and in cancer today in terms of identifying new targets, the right patients to treat, and using, hopefully, the right medicine at the right time. I'm going to talk about the other hot area, in my view anyway, in medicine, which is immune therapies. How can we capitalize on our God-given immune systems to try to turn that power against myeloma? So firstly, here are all the agents that you know, and you've seen, I think, similar slides to this. I'm not going to spend much time on it, but we've been very successful in terms of many classes of drugs, the immunomodulatory drugs here, starting with thalidomide, then lenalidomide, pomalidomide, prototype bortezomib, and then second generation carfilzomib and ixazomib proteasome inhibitors. Kinase inhibitors here don't really have any of these totally FDA approved for myeloma yet, but a new class of drugs. Totally different mechanisms of action here of agents. You heard about the HDAC inhibitors. I'm going to talk about the immune therapies, but this is a team. And you've heard already that 3 drugs is better than 2. The idea is we're not only going to put together multiple drugs, but we're going to use different mechanisms to kill the myeloma cells so that there can't be the development of resistance, and immune therapies are one member of this team. So how does the immune system work? You all know this. You get a cold today and you get better tomorrow. You get an earache and you get better. It's a potent, selective, adaptable system that each of us has been born with. And in fact, it defends us, as this particular slide shows, against infections, bacteria, viruses, and others, but we want to exploit it for its effect and potential effects against cancer. And so there's basically 2 kinds of immune categories you might want to think about, those you're born with and those that adapt relative to the different noxious stimuli that you're exposed to. And so this cartoon slide shows you that we call the innate, and that's the natural system you're born with, immune system, is always ready to try to defend off from foreign invaders, and it is our first line of defense. Over here is the adaptive. So when you're exposed to a new infection or cancer, we hope, then you can have an adaptive immune response such that you can call on those cells with which you were born, your immune cells, to wake up and smell the cancer and do the right thing. And I'll show you that they really do want to reject the tumor. What's been exciting now, after many decades of research, honestly, is that there is the ability now to use your own immune system against cancer. Now, why is it that myeloma cells have been able to evade or escape the immune system? And the issue is that they may have a lot of common fingerprint proteins and other characteristics that don't allow them to be recognized as foreign. They actually are there chronically, in the context of MGUS or smoldering myeloma for many years, and it may be that they can induce what we call tolerance, that the body no longer sees them as foreign anymore. One real sinister thing is when myeloma cells move into the bone marrow, they actually shut off the immune cells that are in your bone marrow, so-called T cells and natural killer cells and others that are just trying to do the right thing, but these myeloma cells can turn them off. And that's turning on other immune inhibiting agents. And in fact, the most common, and I'll mention it here right now, is the expression of, on the myeloma cell, a fingerprint protein called PDL1, and on the immune cells that are in your bone marrow and blood that are trying to recognize and reject myeloma, something called PD1. And when that PD1, PDL1 interaction occurs, it's a break on the immune system, and therapies that take and break that circuit allow those cells then to go forth and do what they really want to do, which is reject cancer. But there's a variety of ways that myeloma can cleverly avoid immune recognition and rejection. Now, cancer immune therapies are in use for other cancers, and that's what this particular slide shows. Monoclonal antibodies, for example, against Herceptin, something called HER2, that is expressed in breast cancer for many years now. There's an antibody that's very effective in breast cancer directed at that fingerprint protein. There are vaccines. There's one in prostate cancer that has been approved. And the checkpoint inhibitors, I just talked about them, but unless you have not been listening, checkpoint inhibitors have really transformed how solid tumors are managed, starting with melanoma and kidney cancer, but now lung cancer. Somebody presenting with metastatic lung cancer, widespread lung cancer, the first treatment that they get nowadays is with these checkpoint inhibitors, antibodies to PD1 or PDL1 that allow the immune system in those patients to react against myeloma. So we in myeloma, it's not for lack of trying, haven't had immune therapies, but that has now changed. So here are the different flavors of immune therapies. In the upper left-hand corner are monoclonal antibodies. So antibodies are medicines that are given by vein, and they go in and they find the myeloma cells in your body. They bind to the myeloma cells by virtue of those fingerprint profile proteins. And then, just like a bacteria or infection would be cleared, then your own immune system can reject the antibody-bound myeloma cell. In the upper right are the overcoming immune suppression class of drugs on the immune team. And those might be like Revlimid and pomalidomide, because we and others have shown for many years that when you take those pills every morning for 21 days, they actually turn on in you, stimulate the T cells and NK cells, natural killer cells, appropriately named, that actually mediate death of your tumor and myeloma. On the lower left here on this slide are what's called CAR T cells, and I'm going to talk about those in detail later, not so much here, but suffice it to say you can program your own immune cells to recognize selectively and kill your own myeloma. And in the lower right-hand corner are vaccines. This is one of my favorite members of the team, because if you think about it, if you get vaccinated against tetanus, you actually then get exposed to tetanus later on, your immune system remembers, and you don't get tetanus. And I think we're not very far away from the time when we can vaccinate patients against their myeloma, and if myeloma tries to grow, your own immune system can remember. That would be really quite impressive and really a nice outcome. Now, here are the types of immune therapies that have been developed and we can talk about here. I've already mentioned monoclonal antibodies. They bind to the tumor cell. They allow your own natural killer cell or T cells to recognize myeloma. They can directly bind here and trigger death circuits inside the cell, or they can bind to the surface of the myeloma cell and cause it to explode. We call that complement lysis. But there's multiple mechanisms. Once an antibody binds to a tumor cell, multiple mediate the tumor cell death. Over here are the CAR T cells, and I'll just mention it now. So here's the T cell that's in all of you, circulating in your blood and in your bone marrow. And what is done is genetically, it is to activate those particular T cells through a T cell receptor. So put a switch on that gets them really programmed to do their thing and kill. But also at the same time, put another antibody, it turns out, binding site, so that now these T cells that are activated bind to something that's on myeloma. And the most common thing is nowadays BCMA, B-cell maturation antigen. But it's a way of getting your own cells to recognize myeloma and kill them. And I've already mentioned vaccines here on the right, and those can either be cellular, or you can take fingerprint proteins from myeloma cells and make vaccines out of little pieces of those proteins called peptides and vaccinate the patient and get a very robust, selective, potent, and even a memory immune response. So the other thing that monoclonal antibodies are useful for, or at least another use, is not only themselves binding to the tumor cell and causing the explosion or letting the natural T cells and NK cells kill the myeloma, they can also be a delivery system. So we and others have used them to, like BCMA or CD38, to bind to myeloma cells selectively, but this time to deliver a poison or an immunotoxin selectively to the tumor. You couldn't use these kinds of immunotoxins or toxins because they are too toxic unless you only designed and made it so selectively they would target myeloma cells. And on the right-hand side is something called a bispecific antibody. And to make that very plain, this is the idea of having one part of an antibody recognize myeloma, say BCMA, and on the other side of the antibody recognize the immune cells. So to put it in plain English, bring your own immune system where it needs to be in your body to the myeloma cell, and hopefully in so doing get a localized immune potent selective response. So antibodies by themselves have many mechanisms to kill. Conjugates bringing poisons to the myeloma cell by virtue of the antibody binding. And then the third thing, bringing the immune system to the antibody-bound myeloma cell. Now, as you know, there is a CD38 monoclonal antibody that is already FDA approved. It's called daratumumab, or Darzalex, as you've heard about previously. The good Dr. Usmani was kind enough to mention that I actually worked on this in the 1980s when I was 13 years old. But in any event, there is an approved antibody that's very effective, and he and many around at this meeting and around the world have utilized this antibody very effectively in relapsed disease as a single agent and in combination. And now, as you've heard so nicely from Dr. Bergsagel, this daratumumab antibody is now being incorporated in initial management as well. I just want to mention here that it's not the only CD38 antibody. One called Isatuximab is another one. And you might say, why would you need another one? Well, it turns out these antibodies bind to different places on the same CD38 target. And we've studied this one in detail, and it has a different mechanism. It has what daratumumab mechanisms of death, but it has additional mechanisms as well. So we don't know, but we are trying additional targeted antibodies against CD38. Here is the early clinical trials that are quite positive in myeloma, showing roughly up to about 60% response rates. And I will tell you that the phase 3 clinical trial, which is like the World Series or the Super Bowl of this particular drug, is finished. It's all accrued. So you will hear sometime in the next year whether we have another antibody called Isatuximab that we can put on our dartboard that Dr. Usmani told you about. I already mentioned this antibody conjugate idea. The concept, again, just so I want to make sure you all get it very clearly, is to use an antibody to deliver a poison, if you will, or a particular drug that can interrupt the growth of myeloma cells. And in fact, this particular one we worked on and published on in 2014. It actually kills the immune way in terms of attracting immune cells, but it also, as shown here, it binds to the myeloma cell. Knock, knock. They think welcome wagons at the door. The myeloma cell opens up, and what do you know? A poison gets delivered inside the myeloma cell. So it's an immune mechanism, and it's a delivery of an immune toxin. And this agent has gone forward. It has FDA fast track now because on this particular slide, about 2/3 of patients' myeloma responds to this, even when the myeloma is not responsive anymore to the IMiDs, proteasome inhibitors, and even the monoclonal antibodies. So very, very exciting immunoconjugate concept. Now, the bispecific antibodies I've already mentioned. We studied, honestly, this particular one, where the idea here is the T cell is an immune cell in the yellow here. That's a natural immune cellular mechanism. It is attracted to the evil target cell, which is a myeloma cell here. And in fact, the T cell, by virtue of something it expresses called CD3, is to the target antigen. We really want to use one called BCMA. But we've studied it, we've published on it. There are clinical trials now ongoing of this so-called bispecific antibody bringing the T cells to the BCMA positive myeloma cell in your body, no matter where that myeloma cell is. So it might be very specific and selective, and maybe the side effects will be less if you can only get the immune reaction where the tumor is rather than more diffusely. Now, talking about vaccines, we've all been vaccinated many times. We've been vaccinated against the flu, which hasn't been a terrific outcome this year. But the idea is that you can induce, as I mentioned earlier, a memory immune response so that if you then get exposed to this stimulus, you will not actually, you'll mount an immune response and you won't get sick with the flu in this example. So in cancer vaccines, we can vaccinate either with cellular, whoopsie daisy, either with cellular vaccines or, as I mentioned earlier, with peptides, little pieces of the fingerprint proteins. Either way, you can vaccinate patients with myeloma and in early stage and in late stage myeloma get an immune reaction in people against their own multiple myeloma. Excuse me. It's a little bit different here if you think about it, because in infections, we actually get vaccinated here on the left-hand side in somebody who isn't sick, so we get vaccinated against the flu before we get the flu, and so hopefully we'll never get it. In cancer, usually we already have cancer, and the idea is then, can we actually wake up the immune system and get it to recognize and do something it isn't doing already, even in the setting of a large amount of cancer cells? So it's a little bit of a harder job to overcome that burden. Now, where have vaccines been used in multiple myeloma? There's a couple of places that they're already being tested where it's really quite exciting. The first one is that in patients who have had a stem cell transplant, after you have had induction with 3 medicines, as Leif and Amrita have told you about, to get ready for a transplant, and you get your transplant, you have low amounts of disease and you get on maintenance therapy. Well, vaccines are typically used after the ablation part, the treatment part, to see if you can get rid of those last few, we call it minimal residual disease tumor cells, in an immune-mediated way. In fact, if you take lenalidomide after an autologous transplant as a maintenance, which is FDA approved, it works, at least in part, by turning on your immune system against your own myeloma at that low dose of 10 milligrams that many of you probably are taking. So why we're so excited about vaccines, and we're not the only ones, is that you could potentially get a selective reaction against myeloma. I already mentioned before, it's very potent and adaptable and selective. And honestly, if you could get an immune reaction, it's more powerful than a transplant. It's way more powerful than any combination of drugs you'll ever get. It probably can get rid of the national debt in terms of myeloma cells. I'm not sure anymore, but it used to be able to do that. In any event, it's an idea that if you had, as I've mentioned, a memory immune response, you could have long-term benefit, and maybe need to get vaccinated at some interval later to keep that benefit, but the idea is very, very exciting. So here are the different kinds of vaccines that have been used in multiple myeloma. The dendritic cell is really, I call it, the welcome wagon of the immune system. It introduces foreign antigens or foreign invaders to your immune system in a way that your immune system recognizes them as foreign. So what's been done in multiple myeloma is to use, these dendritic cells here as welcome wagons against your monoclonal protein, or against, in several studies, or against proteins like BCMA that are actually on the myeloma cell. So introducing and trying to get a response based on a protein or something expressed on the myeloma. The other way that's pretty clever is David Avigan in Boston has taken these dendritic cells and actually linked them, we call it fused them to myeloma cells, and in his vaccine you actually get vaccinated with a myeloma dendritic cell fusion. So now you're introducing the entire myeloma cell as a foreign invader, and you could theoretically get reactions against many, many different foreign proteins on the cell surface. So that's also very exciting, and in very late stages now, after transplantation, MMRF is generously supporting his work in this regard. And then there's what's called a GVAX vaccine. And I'll make this very plain. This is a vaccine that's composed of myeloma cell lines. These are lines of myeloma cells that have been derived from patients a long time ago. But these cell lines are programmed with something called GM-CSF. So what I'm getting to say is these are injecting myeloma cells into patients, and they're giving and putting out this substance called GM-CSF, which is a chemokine. It brings in the immune system. So the vaccine is myeloma, and the vaccine itself is making this GM-CSF that attracts the myeloma, the immune system towards the myeloma, and that's been very effective as well. Early clinical trials in myeloma. Now, in terms of non-cancer-based vaccines, I've already mentioned this several times, but peptides or pieces of the proteins on the cell surface of myeloma have been tried on the left-hand side of this. On the right-hand side, you'll hear from Jonathan in a bit now, but these different peptides or proteins can be mutated. They're changed in myeloma compared to the normal. So in that sense, they offer a specific opportunity to vaccinate patients against their own mutated peptides or proteins. So both of these have gone forward in the past and are very exciting. In terms of other vaccines, I'll just mention them. This is really quite complicated, so I don't want you to get overwhelmed here. But MAGE-A3 is another foreign peptide that has been used WT1 is another foreign peptide. And we have used one on the right-hand side here where we're actually vaccinating patients with smoldering myeloma long before they're getting sick. And we can show that you can get a memory immune response in patients with smoldering myeloma. And wouldn't that be a happy occasion if you could get an immune response in patients before they ever get need for treatment, and maybe someday prevent them from ever evolving towards active disease? Now, I want to finish up with a discussion of cellular therapies. Cellular therapies are based, as I will mention here, on T cells, those natural immune cells with which all of us are born. And in fact, the idea here is that you can program somebody's own T cells, harvest them from the body, and then do things to educate them outside of your body to recognize selectively multiple myeloma. There are several different ways, but the main one that you've heard about are CAR T cells. And let me make this very simple. So you go in and you get T cells removed from your blood in a process called pheresis, the same kind of process that's used for people to donate platelets when they're trying to help for transfusions of kids and adults with cancer. When you had your stem cells collected, if you had a transplant, it's the same process. This time the T cells are removed, and then they are infected outside of you. They are educated or programmed actually to recognize a certain target, most commonly BCMA in myeloma, and they're programmed at the same time to activate the killing mechanism in those T cells. Then they're grown up in large numbers outside of your body and transfused back, and I like to call it your own immune army against your own myeloma. And that's called CAR T cells. The other kinds here, I'll just mention quickly in passing. T cell receptor engineered T cells are ways to manipulate that T cell switch in your T cells to turn them on against myeloma. And then Dr. Borrello at Johns Hopkins has talked about what's called marrow-infiltrating lymphocytes, MILs. These are the cells that, if we do a bone marrow, these are the immune cells that are in your bone marrow trying to get those evil myeloma cells. But as I mentioned very much earlier, the myeloma cells hide from those cells and they shut them off. But MILs are another mechanism. Maybe there's something special about those cells that are already near the myeloma. So finishing up with the CAR T cells here, I already mentioned this, but there's just in cartoon fashion. When you have a natural switch here that turns on T cells, it is changed in the concept of a CAR T cell by putting the switch in the on position. So these T cells are now ready to go, but also getting it so they only go to one place. And in myeloma, it's in fact something that's built in and binding to BCMA, or the target that's pretty selective only on normal plasma cells and myeloma. So here, I've already mentioned this about pheresis and constructing the cells and reintroducing them, so I won't mention that. But what I will mention is the several studies that have been done. And I got to be very careful what I say about CAR T cells. I've learned this very recently. But honestly, I think it's hugely promising and very exciting. But please hear me, it's also very early. Much work left to be done in terms of making them more potent and selective, maybe getting memory T cells, and in particular, making sure that they can survive long enough to do their work and have fewer side effects. So we're in the very early days of CAR T cells. It's hugely exciting, but I think it's now in protocols not to be used before one has used many of the other already approved agents. Nonetheless, here are the results so far. So one trial is called B2121. It's the Bluebird trial. 90% of people who had far advanced myeloma had a response, which is quite remarkable. And in fact, the idea is that the toxicity, which is called cytokine release syndrome, CRS, is now being better controlled. In plain English, if you grow up a whole army, your own immune army, and then you give it back it'll go forward and get the myeloma. But part of the way it works is it's been turned on and it further stimulates these cytokines in you. So you get high fevers and blood pressure fluctuations, and even swelling around the brain has been a problem. It's called cytokine release syndrome, and we use antibodies to interleukin 6, for example, or IL-6 receptor to control this. But it's early days, very exciting, 90% response rate in far advanced disease. Here is the University of Pennsylvania team, together with Novartis. 11 patients had at least a partial response. Here's the NIH team. 9 of 11 patients responded. And the idea here is, you've heard about minimal residual disease from others, but you can get down to really impressive responses. Fewer than 1 myeloma in 100,000 or even 1 million myeloma cells. And I'll talk about MRD in a few minutes later. And then we have to watch out for China, as you may have heard from Washington. But in any event, China is trying to help us by making CAR T cells. And in fact, this particular protocol was very exciting. This time they have CAR T cells that target BCMA and another target called CD19 at the same time, trying in so doing to make them more selective. And they had very high response rates reported at ASCO and at ASH. And then the Memorial Sloan Kettering, very small experience, but they're doing all kinds of clever maneuvers to make the CAR T cells better. So the summary for CAR T cells is, it's taking advantage of your own T cells, educating them outside of your body, transfusing them back after they've been expanded, and remarkable progress, but early days. And then this is my favorite, actually, and I spoke of this at our blood bank talk recently. But I think in the future what's going to happen, and I don't think the future is that far away, is we're going to get normal donors to come in and donate their own T cells, just like they come in now and donate platelets to help kids and adults with cancer. They're going to donate their own T cells, and then we genetically are going to take out the ability of those T cells to recognize patients as somebody else or foreign. And we're going to, at the same time, make those T cells put in the education to recognize BCMA, for example, and then put that bottle of programmed normal donor T cells on the shelf. So that when somebody comes in with new myeloma in the future, my opinion is we'll reduce their myeloma with 3 or 4 drugs, as Dr. Bergshagel mentioned. And then I think once this is made safe, we'll say, I'll take that bottle of the BCMA allogeneic CAR T cells, please, and you'll just get an infusion of those T cells. And wouldn't that be special? It would be really neat. And I don't think it's Star Wars. I think it's coming. So what I've tried to say here is that CAR T cells are very exciting. I have tried to urge caution. It's excitement, but there's much work left to be done. I have mentioned to you that we need to use BCMA as the best target right now. There are other targets that are being investigated. And I mentioned to you that this idea of antibodies by themselves or antibodies as a delivery system to bring in a poison in an immunotoxin or to bring in your immune system in a bispecific way, bring in the immune cells just where you want them, are now under clinical investigation. So I've covered an awful lot of material, but the summary of it is that, in fact, we were born with, each and every one of us, an immune system that is awesome. It couldn't not have been possibly designed by anybody other than somebody upstairs. The point of this is it's potent, selective, and adaptable. And if we can harness that immune system, and I believe only if we can harvest that immune system, will we actually have long-lasting, durable responses. We can get to minimal residual disease with transplants, drugs, and other things, but we need to keep it that way if we intend to really cure patients. And I think what I've tried to say to you, there's a variety of immune ways where I think we can restore the ability in you as a patient, your immune system, to recognize and reject myeloma and keep it away. Anyway, thanks very, very much.
Ken, just stay. So there are a lot of questions about CAR T cells, Ken, and a lot of excitement. And I think the perhaps the most important one is, Is CAR T therapy available outside clinical trials, and when will CAR T therapy be available?
Yeah, so that's a super question. The first part is easy. No, it is not available outside of a clinical trial. There are more and more trials coming, but it really should only be done in the context of a clinical trial. I've mentioned to you the process, but the idea is that you can get pheresis, you get infection, and the infection or the education of the myeloma cell is done by different companies different ways. Most of them are educated to recognize BCMA, but there's other differences on how they get them to proliferate and what kinds of cells. Should we have the memory cells or should we have the killer T cells? What kind of cells do we really want in the CAR cells? And there's a lot of work going on in terms of how we can get them to survive. See, in the kids who get CD19 CARs for recurrent leukemia and they apparently are cured, those T cells persist. But in adults, we haven't really figured that out yet. So the idea is that it needs to be done in a clinical trial, and it's only in patients who really have exhausted pretty much all other therapies. The other thing that we as a world have to figure out is they cost about $450,000 per product, not counting the couple $100,000 more that is needed when you go in the hospital to have them administered. So I think there's a huge financial toxicity that also needs to be addressed, and I hope and I expect that we will.
Okay, last question then. Can you take things like vitamins or immune-boosting foods to build up your immune system?
Yeah, I think the answer is yes. I think that, I always like to say this, but keeping yourself in the best shape possible, physically, mentally, spiritually, etc., is my opinion, and we heard beautiful talks this morning, but I think it's more than 50% of the battle. You will tolerate therapy better, your immune system will be better, so that I do think that there is, in fact, that is very good advice. I'm not sure there's a particular agent or a particular immune stimulant that I would recommend, but I think good nutrition is absolutely key. And I would add one other quick thing. You know, when you think about, as you've heard this morning about MGUS and smoldering multiple myeloma, and then it gets to be myeloma, so how does that happen? We don't know for sure, but the genes change, as Leif and others have shown, inside the cell. There's more ability to grow and proliferate, but there's also a decrease in the immune function in the host. So keeping healthy with good nutrition, good lifestyle, et cetera, can keep your immune system better, which may very well be important in keeping myeloma early stages, at least for a longer duration.
Thank you, Ken.
Okay.